Welcome to the Fatty Acid Forum, a video series on the art and science of feeding fatty acids to dairy cows. In this episode of the Fatty Acid Forum, join Dr. Charlie Staples from the University of Florida as he begins a series on the essential fatty acids by exploring the question, which fatty acids are dietary requirements and where can they be found? We hope you enjoy this episode of the Fatty Acid Forum, sponsored by Virtus Nutrition. And now, here's Dr. Staples.
Long-chain fatty acids are considered essential dietary nutrients for all species of animals, yet we don't formulate diets for dairy animals to specific daily amounts of intake of these essential fatty acids, at least not yet. The purpose of this first presentation on essential fatty acids is to identify and describe those essentials, explain what makes them essential in the diet, discuss which feedstuffs are good sources of these essential fatty acids, and explain how the general intake of these essential fatty acids by lactating dairy cows can vary widely. Now, for years we've known that crude protein is composed of individual amino acids. Several of those amino acids are considered essential and must be included in the diet in order to meet the animal's requirements. Essential amino acids would certainly include methionine, lysine, and others. But in general, we've paid less attention to the individual fatty acids that compose crude fat and to what their role is in animal metabolism. The common long-chain fatty acids include palmitic, stearic acid, oleic acid, linoleic, and linolenic acid. For a nutrient to be classified as an essential nutrient, it must be supplied in the diet because the animal's tissues are incapable of synthesizing the required amounts. The two long-chain fatty acids that have been identified as essential are linoleic acid and linolenic acid. I have the scientific terminology listed here as well. Linoleic acid is abbreviated C18-2 and is classified as an omega-6 fatty acid. Linolenic acid is abbreviated C18-3 and is classified as an omega-3 fatty acid. And that nomenclature is further explained in the following slide. So at the top is the chemical structure of linoleic acid. Capital C here in C18-2 refers to the carbon atom, and there are 18 carbon atoms in the chemical structure of linoleic acid, and they are numbered 1 through 18. All of the carbons are connected by a single bond, with two exceptions. There's a double bond that connects the 9th and 10th carbon here, and again between the 12th and 13th carbon. In the 18-2 terminology, the 18 refers to the 18 carbons, and the 2 refers to these two double bonds. The omega-6 phrase is a further classification of this fatty acid that indicates that the first double bond occurs at the 6th carbon when counting from the methyl end of the carbon chain. So we have 1, 2, 3, 4, 5, 6. Other fatty acids can belong to the omega-6 family, which we'll look at briefly later. To complete the story of how fatty acids are described, I've included a cis-9, cis-12 octadecadienoic acid. The cis term refers to the configuration of the carbon atoms, and the number 9 and the number 12 refer to the location of the double bonds when counting from the opposite end of the carbon molecule. So going from right to left, double bonds are at the 9th and 12th carbon atom. Lastly, the words octadecadienoic acid is a Greek nomenclature in which octadeca refers to 18 carbons and DIE refers to the two carbon bonds. So likewise, linolenic acid has 18 carbons, but it has 3 double bonds, with the first 2 double bonds being in the same location and the third double bond between the 15th and 16th carbon. So again, thus the C18-3 is the 18 carbons and the 3 double bonds. Linolenic acid is classified as an omega-3 fatty acid because its first double bond occurs at the 3rd carbon from the methyl end. And using the Greek terminology, octadecatrienoic acid Notice it is a tri rather than a dienoic acid because it has 3 double bonds instead of 2 double bonds. Again, other fatty acids are classified as omega-3 fatty acids, which we'll talk a little bit about later. And omega-6 and omega-3 fatty acids have different functions in tissues, which we'll get into in another webinar. So linoleic acid at the bottom contains the word oleic acid because it is derived from oleic acid. And oleic acid also has 18 carbons, but it only has 1 double bond here between the 9th and the 10th carbons. There's an important enzyme called delta-12 desaturase enzyme, which can insert this second double bond between the 12th and 13th carbon. When it does that to oleic acid, linoleic acid is formed. And this enzyme is found in plants, but not in animals or in bacteria, and this is critical. So neither animals like the dairy cow nor the bacteria living inside the rumen of the cow can synthesize linoleic acid, so the animal must consume vegetation in order to have linoleic acid in their diet. Likewise, the delta-15 desaturase enzyme, found again only in plants, can insert a double bond between carbon 15 and 16, and again not found in animal tissue or bacteria, so linolenic acid must be supplied to the cow through the consumption of vegetation. Since nearly all of the feeds consumed by cows are of plant origin, cows are always consuming some linoleic and some linolenic acids. They are of course consuming other long-chain fatty acids as well, such as oleic acid, which is 18 carbons and 1 double bond, stearic acid of 18 carbons and no double bonds, and palmitic acid, which is 16 carbons and no double bonds. And the dietary forages, like alfalfa, ryegrass, which are in this green forages group, and corn silage, contain low amounts of fatty acids, maybe 1 to 3% of the total dry matter, which isn't much. But the good news is that the majority of the individual fatty acids found in these feedstuffs are the essential fatty acids. So green forages from temperate growing regions contain much more linolenic acid than linoleic acid. You can see in this case around 60% linolenic and only 13% linoleic. Forages grown in tropical areas of the world, such as Bermuda grasses, contain slightly more 18.2 than 18.3. Next, the cereal grains, such as corn and barley, also have low concentrations of fatty acids, only 2 to 3%. But again, they're high in the essential fatty acids, or specifically linoleic, not much linoleic, but 42 to 40 or 68, 42 to 68% linoleic acid, just depending on the grain source. The oil seeds, such as soybeans and whole cottonseed, contain more fatty acids. You can see they're about 20% fatty acid. So they're more than the cereals or the forages. And these are very good sources of 18.2. Over here you can see they're 54% linoleic acid. Because they're high in oil or fatty acids and high in linoleic, they are excellent sources of linoleic acid. The one oil seed that is an excellent source of 18.3 is flaxseed, which is also called linseed. It's about 25% fatty acid and about 55% linolenic acid. So it's one of the few excellent sources of linolenic acid that we have coming from vegetation. Fat sources are often included in diet as an energy source, and so a couple of these I've got listed on this table are tallow and yellow grease. You can see they're very high in fatty acids, about 90% fatty acids, but their main fatty acid source is oleic acid. This you can see they're in their mid-40s, 41 to 44% oleic acid, and much less linoleic, and even lower in linolenic. So as we put diets together for our dairy cows composed of these different feedstuffs, the diets are going to have varying concentrations of the individual fatty acids. But another factor that influences the concentration of linoleic and linolenic acid in the diet are how those particular forages are stored. When corn plants represented by the orange line and sorghum plants represented by the blue line were ensiled, the concentration of C18-2 did not change very much. So looking at this first graph over here where the plant stored in the silo up to 160 days, you can see that the concentration of linoleic acid is not changing very much. It's around 36 to 40%. However, the concentration of 18-3, which is the graph here on the right, is decreased for both corn silage and sorghum silage, with the decrease being greatest in sorghum silage, going from about 20% here at the time of ensiling down to close to 13% after 160 days of ensiling. So in addition, the concentrations of these essential fatty acids can decrease a few percentage units if 1, the forage is wilted prior to ensiling, 2, if there is a delay in sealing of the silage, and 3, if the silage is allowed to heat up after the silo is opened. All of these practices can result in the oxidative destruction of these unsaturated fatty acids as the fatty acids are exposed to oxygen. And 18-3 just seems to be more sensitive to oxidation than 18-2 because of the more double bonds it has. Storing forages in the form of hay can have a much more negative effect on the fatty acid concentration of the forage compared with ensiling, and that's shown in this study here. Workers from France analyzed the fatty acids in alfalfa samples at harvest, called fresh here, which is the blue bar, and they also looked at the fatty acids of the alfalfa as they made it into silage, which is the orange bar, and after they made the alfalfa into hay, which is the purple bar. So drying the alfalfa reduced the fatty acid content from a bit over 2% down to 1%. The concentration of 18-2 was not affected very much by either making the alfalfa into silage or hay. You can see it's all here around 17 to 18%. But the concentration of C18-3 was dramatically decreased when the alfalfa was made in the hay, going from about 40% here down to about 15%. So making alfalfa into hay would markedly reduce the concentration of 18-3. And if we multiply the fatty acid content of the diet of hay, which is 1%, by the concentration of 18-3 as a percent of the total fatty acids here at 15%, we could see that the concentration of 18-3 would decrease from about 0.85% in fresh forage down to about 0.15% of the dry matter when it's made into hay. Because the dietary ingredients can vary quite a bit in essential fatty acid content, intake of essential fatty acids can also vary widely. So I've listed 3 diets composed of typical feedstuffs along the left-hand side. Diet 1 contains feedstuffs that are relatively low in essential fatty acids such as alfalfa and barley. And we don't have any supplemental fat sources in Diet 1. Diet 2 contains more corn silage and less alfalfa silage compared to Diet 1. In addition, we've replaced the barley grain by adding some corn. We've added an animal vegetable fat into Diet 2. Diet 3 has all corn silage, no alfalfa silage. We've also included in here the corn, no barley, and lastly included some whole cottonseed as a fat source. And we entered these feedstuffs using the NRC software to create these diets in which dry matter intake is at 56 pounds a day, and this resulted in milk yields of between 90 to 95 pounds. These two bar graphs show the intake of total fatty acid and the intake of the individual essential fatty acids. The graph on the left is the intake of all fatty acids, and they range from about 500 grams a day for diet 1 up to over 1,200 grams a day for diets 2 and 3, which contained either an animal-vegetable blend, which was diet 2, or whole cottonseed, which was diet 3. So we've more than doubled the intake of fatty acids by adding these supplemental fat sources. The intakes of the essential fatty acids are shown on the graph on the right. The intake of 18.2, which is the orange bar, was as low as 182 grams per day for diet 1, going up to 686 grams per day for diet 3 due to the high concentration of C18.2 in whole cottonseed. Intake of 18.3, which is the blue part of the bar, was always less than the intake of 18.2 across diets and is the typical situation in the field. Intake of 18.3 decreased from 99 grams in diet 1 down to 34 grams in diet 3. And this change was largely caused by removing more and more alfalfa from the diet and replacing it with corn silage. So as you can see, the intake of essential fatty acids can be dramatically altered by changing the dietary formulation. Well, these two essential fatty acids serve as substrates to synthesize other very important long-chain fatty acids, which will be discussed in future webinars. But C18.2 is elongated by 2 carbons and desaturated by animal tissues. This is done by adding 2 carbons. This forms arachidonic acid, or C24 double bonds. This is another omega-6 fatty acid. An arachidonic acid is not typically found in feedstuffs, but is a very common fatty acid found in animal tissues. 18.3 can be elongated by 2 double bonds and desaturated to form C25 eicosapentaenoic acid, abbreviated EPA, and this in turn can be converted to C22,5 docosapentaenoic acid and to C22,6 by another desaturation step to form docosahexaenoic acid, DHA. All of these are omega-3 fatty acids, and EPA and DHA are found in feeds coming from marine sources such as fish oil, fish meal, and marine algae. Note that a fatty acid in one family cannot be converted into another family. So once an omega-6, always an omega-6 fatty acid. In summary, linoleic and linolenic acids must be in the diet of dairy animals to meet their requirement, and they serve as parent compounds for other long-chain fatty acids. These are the only 2 dietary essential long-chain fatty acids. The fatty acids of cereal grains and oil seeds are enriched in linoleic acid. However, only oil seeds are excellent sources because of their high oil content. Fresh or ensiled temperate grasses and linseeds are excellent sources of linolenic acid. Fourthly, forages lose much of their fatty acid and linolenic acid content when made into hay, but losses are small when ensiled. And last, intake of essential fatty acids by dairy cows can vary widely.